Hello, Biology 230. This is Dr. Chastity Bradford, and I will be your tour guide as we tour the cell. This is Module 4, and Module 4 covers both Chapters 6 and 7. And you'll notice that these chapters are divided into key concepts. So this is key concept 6.1, noted as Chapter 6.1. And on the background of this slide, you'll notice that I have a lung tissue. This is a immunohistochemical stain, which is a technique that we use when we take tissues and we stain those tissues and we look at various cells. And so this is a lung tissue, human lung tissue, from a patient who had idiopathic pulmonary fibrosis, which is just a disease of the lung. And what you'll notice in this lung sample is that there are various blood vessels noted here, because the inside of these blood vessels is called the intima, and it stains green. Those cells stain green. These are endothelial cells. And also you'll notice some orange staining here, and there are various cells that stain orange, but in this tissue you'll notice that around the intima there is a media layer that I'm highlighting. This media layer of the vessel has smooth muscle cells, and so the smooth muscle cells in this blood vessel are staining orange. And so this just illustrates what we've learned previously in the previous chapters about the hierarchy of organisms. And you notice that in this tissue then is made of cells. Okay, and there's various types of cells. Although there are many different types of cells, there are a lot of commonalities and common features that cells share. And that's what we will examine in this chapter. And in chapter 6.1, what we're going to discuss is the tools and the techniques that researchers and students like yourself who are conducting research with us that we use to help us understand cells. In order to study cells, we use microscopes, of course. Why do we use microscopes? Because we can't see a lot of things with our naked eye. They're just too small. And so that's all this slide is showing us, is that we need microscopes to see the things that are too small, right? Duh. But now we're going to go into the different types of microscopes. So one of them that we're going to discuss, of course, is a light microscope. This is the one that most of us are really familiar with. It uses visible light in order to magnify images, okay? And the quality of that image is going to depend on three things: magnification, resolution, and contrast. And what you have here, that is figure 6.2 in your book, demonstrates what we can see with our eyes, unaided eye, what we can see under a light microscope, and what we may need an electron microscope for. And so what this just shows you is that with the unaided eye, or the naked eye, you can see just a person standing next to you, or your professor like myself. You can tell how short I am, or how tall I am, for that matter. You can look at frog eggs with the unaided eye. But if you wanted to really look at a cell, if you wanted to look at a cell, whether it's a plant cell or an animal cell, you really need a light microscope to do that. And if you wanted to look at the details of the cell, the subcellular structures, you need an electron microscope. And in addition to that, there are some techniques that you'll need to utilize if you really wanted to study the subcellular structures, and that's what we'll discuss today. Most of these subcellular structures, including organelles, and organelles are membrane-bound compartments, or membrane-enclosed compartments, if you will. Most of these structures are just too small to even be seen with a light microscope. So there are other microscopes that we use, and two of them are electron microscopes, and two types of the electron microscopes are scanning electron microscopes and transmission electron microscopes. Scanning electron microscopes, or SEMs, both of them both scanning and transmission use beams of electrons to provide us images. Now, the scanning electron microscopes, they focus in on the surface of a specimen, and then we're able to kind of see them in 3D, okay? And I'll show you images of that next, with the next slide. And transmission electron microscopes, on the other hand, with these microscopes, we can see the internal ultrastructures, okay? And there are disadvantages to using these electron microscopes versus light microscopes. And one of them is that with these electron microscopes, we can only visualize dead specimens, okay? With the light microscopes and some others, you can actually look at living organisms as they move. And that's one of the advantages of upcoming technology that exists now. Okay, that we can see cells and we can see interactions with other cells. And so research in biology is just that exciting. Okay, so now what I'm showing you are images from some manuscripts that researchers have produced. And this first one here, 26, 27, 28, and 29, in this figure, you can begin to see some of the internal structures, right? Some of the internal cellular structures you can see here. Now, you would not be able to see this with a light microscope, nor would you be able to see this with the unaided eye, okay? So I want you to tell me what's the difference between the image produced here and the image produced here. Can you tell the difference? The images produced in 1, 2, 3, 4, 5, and 6 are very detailed, right? Almost like 3D. That's what we mentioned before. It's almost like the 3D. And so we can visualize the surface of these cells, the surface of the internal structures of these cells, okay? So if you go back, you'll notice that the scanning electron microscopes provide images that look 3D, surface. And the transmission electron microscopes provide images using these electrons of internal ultrastructures. So this would be what type versus this, okay? Now, there are also a few firsts as it concerns the cell. Robert Hooke, he was the first to look at dead cells through a microscope as far as we know. Now, Leeuwenhoek was the first scientist to visualize living cells. And so I think they would be really fascinated to see how far we've come in terms of moving from light microscopy to these electron microscopy, okay? Scanning and transmission. And it's important that you understand the difference between the two, because we will give you images and ask you which one is scanning, which one is transmission, and what's the difference, okay? Scientists, we use microscopes, and as the scientists began to use microscopes over the years, they began to find some common themes among cells, among organisms, okay, that had cells in them, common themes, okay? And the common theme, the term that we use now is the cell theory, okay? So based on all the things that they found that were common in each organism, the cells that they found in every organism they examined, they all came up with these four main ideas. One, all of the organisms that they studied were made of cells. They noticed that. They noticed that the cell is the simplest collection of matter that can live. They determined that cell structure correlates with cell function. And this is something that you really want to remember. Structure correlates with function. And also, another idea is that cells arise from pre-existing cells. So there are basically four main ideas for the cell theory. All organisms are made of cells. The cell is the simplest living thing, if you will. That the cell structure is related to its function, and that all cells arise from pre-existing cells. And there are other ideas about the cell theory, but these are four that it's important that you understand. Now, how do we truly study the cells? Okay, we look at them and we can say, okay, this is what they look like, but how can we study their function? So in order for biologists to study the function, we have to isolate organelles. And how do we do that? We use different techniques and tools to isolate the organelles. And one of the techniques that's used is called cell fractionation. Cell fractionation lyses or takes the cells apart, and then it separates the major organelles from one another. How is it done? What tool is used? The ultracentrifuge is used to fractionate cells. And this allows scientists to determine the function of the organelles. So structure is correlated to function, and in order to ascertain what or determine what function correlates to what structure of the cell, cell fractionation is used. And this is how it's done. For instance, let's say we take that lung tissue from the idiopathic pulmonary fibrotic patients. We homogenize or blend, if you will, like a blender, homogenize or blend up the lung tissue. And then you come up with this, what is called a homogenate. And then using an ultracentrifuge, you spin down this sample, and what you end up with is a supernatant and also a pellet. And in this case, the pellet now has nuclei and cellular debris, and then you end up with supernatant. Okay? Then you spin this down again, and what it does is it continues to separate and fractionate. And what you have now is supernatant again, but you also now have a pellet that is enriched in mitochondria. And if these were plant cells, you'd have chloroplast in the pellet. So now if we centrifuge again, so this is a process of differential centrifugation, we centrifuge again, and now we have a pellet that has microsomes, and we have our supernatant. So if we take that and we spin it again for 3 hours at a certain G force, you notice that now we have a pellet that has ribosomes. And so at each step, we're able to isolate different organelles. Here, mitochondria, here, microsomes, and at this step, ribosomes. And then we as scientists and the students that are studying, working with us, are able to study these organelles and their function. This slide just shows you what an ultracentrifuge looks like, and it lets you know that it can spin very fast. So it can spin at 130,000 revolutions per minute and apply forces of more than 1 million times gravity. And what this shows you, demonstrates here, is a rotor. This is a rotor. So each of those samples, like we saw before, are placed in what's called a conical tube. These are conical tubes with these blue tops. And they're placed into this rotor. It's balanced. And then this is placed in here into the centrifuge. And they just spin it down. And then take out what they need, study it, spin it again, take out what they need, and study it every time that they change the speed, change the G-force. They're able to isolate a different organelle and study that organelle's function within that cell. And that concludes chapter 6.1.